In the last part of the lecture, we discussed about the antibodies and an overview of the adaptive immune response. Now, continuing on with adaptive immunity, we will now be discussing about the antibody-mediated response. So, sometimes we call it the humoral response because it produces humors, or basically, because it's the antibody which is when we talk about humors, it's more of an archaic term used in around the Middle Ages, the Renaissance and pre-Renaissance period, to describe some fluids, humors of the body. So anyway, when we talk about humoral response, right now, this is the modern definition. B cells triggered to make antibodies specific to a particular pathogen or toxin. So we had in the previous discussion before we end, we saw that how the cells actually interacts with an antigen. So you have the cells engulfing the bacteria and breaking them down and taking parts of that bacteria and presenting them outside. So the B cells also does that. So they engulf the pathogen, the antigen, and then they create antibodies that are specific to that pathogen or toxin that they engulf. So antibodies bind to the antigenic particle, facilitating its uptake by phagocytic leukocytes. By the way, the antigen is not only limited to the viruses, the viral particles, the bacteria, but it can also be used against toxins, just like a spider venom, etc. So bound antibodies can keep pathogen from infecting other cells. So one of the ways, because antibodies can bind the pathogens or the toxins, so that's why antivenom are made up of antibodies as well. They actually prevent the toxins, the pathogens from actually diffusing throughout the rest of the body, so they keep them in place. So here is a view of your antibody-mediated immune response. So starting off, we have your naive B cell. So the naive B cell becomes activated due to the actions of the complement system as well. So the complement system signals that, oh, there is something happening here, red alert, and then they will go there. And then they will be exposed to that pathogen. And then you have your dendritic cells, which we also have your dendritic cell here. The dendritic cells engulfs and digests the same kind of bacterium that the B cell encountered. So the bacterial fragments bound to the MHC markers becomes displayed on the surface of the dendritic cell. So the dendritic cells displays markers from that pathogen. For example, here is the bacteria. And then you have here receptors of the naive helper T cells recognize the bacterial antigen displayed by the dendritic cells. So the dendritic cells basically is the one that helps in identifying these pathogens, and it tells them that you should this is the person, or rather this is our enemy, look for it. Okay, so that's our naive helper T cell. So then when it becomes exposed, when the naive helper T cell becomes exposed to it, so exposed to the antigen presenting dendritic cells, it then divides into memory helper T cell and an effector helper T cell. So the receptors of one of the effector helper T cells recognize and bind to the antigen displayed by the B cell. So you have the effector T cell, and then it will see the activated B cell that was exposed to the bacteria from number 1 here. So basically, your naive B cell, once it becomes exposed, no longer naive, it's become the activated B cell. And so this is the one the effector helper T cells meets. So the effector helper T cell will release cytokines. So the cytokines will induce the B cell to undergo repeated mitotic division. So the activated B cell will now divide. And this activated B cell the divisions will be divided into a memory B cells that will be retained by the body for a long time as a memory of that antigen, or the memory of that bacteria. And an effector B cell, where the effector B cell is the one that will release the antibodies against that particular pathogen or bacteria. So basically, this is the antibody-mediated cell response. So you have the B cells and T cells helping one another. Sorry. Okay, so next slide. So here is your dendritic cells that is engulfing your pathogen. And then you have here an immunological synapse. So this is a zoning in point of contact between the T cell and the antigen presenting cell. So basically, this is how the antigen presenting cell tells the naive T cell that this is your target. So they have in their cell surfaces, they have proteins in their cell surfaces that basically not exactly touching, but that encounters one another, and it will identify this is my target. So basically that's that. So it's like touching one another, but not exactly touching. So the antigen binds only to the matching B cell receptor. So that's one thing about the adaptive immune response. They are very specific. So once the antigen binds the matching receptor, that B cell binds that binds this antigen undergoes the mitosis. So this is so that only the B cell, this is in the memory, clonal selection, this also applies for the memory cell, so that only the correct B cell with the correct memory becomes activated whenever we encounter that antigen. So only the B cell that has the correct or the matching antibody to the antigen, the matching receptor to the antigen will become activated. So it needs the antigen to be actually present and to bind to that correct B cell, and that B cell will then be activated, and it will create multiply to form the effector B cells as to fight against that antigen. So this is also a mechanism of our adaptive immunity. So only the correct. So basically, for example, if you have chickenpox, so only your antibodies, or rather your B cell that has the memory for chickenpox, will become activated once you get exposed to another chickenpox virus, and not the antibody for flu, because it follows a different pathway of response for our cells. So that means it's actually adaptive. It's tailor-made for a specific disease or virus. So active immunization. The vaccine elicits a primary immune response, and you have the second vaccine dose, which is usually the booster shot, which elicits a secondary immune response. This is what you call active immunization, whereas the immunity comes from activating our cells, activating our immune responses, as opposed to the passive immunization. So instead of vaccines, we receive antibody purified from someone else's blood. So we receive other people's antibodies. So this immunity is short-lived because it only lasts as long as the injected antibodies do. And proteins inside our bodies really doesn't last long. So this lasts days to weeks, but only that. So a carbohydrate called the H antigen on the red blood cells, blood cell membranes, determines the blood type. So we have, this is actually one of the ways where we have the blood typing. Okay, before I actually move on to this one, let me go back to this passive and active immunization, because in the COVID response that we had, if you have heard of COVID, the patients that survive COVID, they donate their blood sera to the patients, to the current COVID patients. This is a form of passive immunization. Their blood serum still, because their blood serum still contains the antibodies against COVID. Because of course, since they got sick and they got well from that sickness, so that means their body has been able to successfully mount a response. So since they just recently got well, they still have remains of those responses inside their blood. So they donate their blood that contains the antibody against COVID, and they don't the blood will become purified, so they remove the external cells, leaving only the plasma, which contains your antibodies, and that will be given to the COVID patients to help those patients fight off COVID. So that is a form of passive immunization. Whereas for the vaccination program is a form of active immunization, because we are not given the antibodies, we are actually given proteins or RNA to make the proteins of those of the COVID virus, so that our own bodies will be the one to produce the actual antibodies. So basically those are that. Okay, now aside from the functions against diseases, so you have your blood typing. So you have a carbohydrate called the H antigen on the red blood cells, determining the blood type. So you have two forms, A and B. So this is one of the most common blood typings, blood type A and the blood type B. because actually ABO blood type A, we have 4 groups: A, B, AB, and O. But in reality, there is only 2 types of antigens, the A and the B. So the 4 groups just depends on having the presence or absence of one or both antigens. So if you transfusion of an incompatible type causes an immune response that ruptures blood cells, releasing dangerous amounts of hemoglobin in the blood. So that's why blood typing is necessary, so that the correct blood type will be given to the patient that needs blood. Otherwise, your body will attack the foreign blood. So here is your ABO blood typing. So basically, blood type A has AH antigen on their blood, and they contain anti-B antibodies. And then you have the blood type B that contains the BH antigens, so they contain anti-A antibodies in their blood. So that means their antibodies is against the other blood type. Now for the blood type AB, they have both A and B. So they do not have antibodies against either antigen because our bodies do not want to attack our own cells. So basically, the blood type AB are the universal acceptors of blood type. So they are the most safe, or rather, they can accept blood type A, they can also accept blood type B. Now, we have the blood type O that has neither A nor B blood type, but their antibodies, they have both antibodies, anti-A and anti-B. That is why blood type O is called the universal blood donor, because since they do not have the H antigen on their blood, they can donate to blood type A, blood type B, and blood type AB, as well as, of course, the other blood type O. But they would have difficulty receiving blood because the only blood that they can receive needs to come from blood type O individuals. So that is blood typing, blood transfusion, and basically the antibody requirement for that. Now moving on, the cell-mediated response of the adaptive immunity. So for the antibody-mediated response, we talk about the B cells and the antibodies. For the cell-mediated response, it's actually mostly dominated by the T cells. So you have cytotoxic T cells and other lymphocytes that are produced. So they recognize intracellular pathogens. So that means these are pathogens that are inside the cells. So antibodies here is not necessarily involved. So we also have natural killer cells. So natural killer cells, or the NK cells, they can kill cells that lack MHC markers. So basically MHC, major histocompatibility complex. So this is somewhat like an ID. Basically, if you are in an organization, you need to have an ID to enter this restricted building. So it's like an ID. So people with no ID are immediately caught by the security guard and asked to produce a proper identification. Otherwise, they would be taken off the building, or anyway. So the natural killer cells are activated by cytokines. So here is your cell-mediated response. So again, it also involves dendritic cells. So dendritic cells, first, you have dendritic cells engulfing your virus-infected body cells. So take note, this is our own cell, but this one is infected by a virus. Because the cell-mediated responses, it majors. It actually, most of its action is against cells, our own cells that have been compromised, that either became cancerous or has been infected with the virus. So your dendritic cell will then engulf the suspected virus-infected body cell, and then your the receptors on the naive cytotoxic T cell binds to the viral antigen displayed by the dendritic cells. So this type of interaction will then activate your helper T cell. So the receptor on the naive helper T cell bind to the viral antigen displayed by the dendritic cells. So the interaction 2 and interaction 3 here in numbers 2 and 3. So in number 2, you have here cytotoxic receptors on the naive cytotoxic T cell binding to viral antigen displayed on the dendritic cells. This interaction will activate your cytotoxic T cell, leading to the differentiation of this activated cytotoxic T cell into a memory cytotoxic T cell and an effector cytotoxic T cell. So for the other interaction with the dendritic cell, another naive, this time it's for a naive helper T cell. So we have a cytotoxic T cell and a helper T cell. So for the naive helper T cell, so once it binds also to the dendritic cells. So the activated helper T cells then divides again, and its many descendants mature as effector and memory cells, each with T cell receptors that recognize your virus. So basically, the naive cells in the adaptive immunity, it actually both antibody and cell mediated, they must be first they were at first naive, and then they become activated. The reason for that is because they need to recognize first who they will fight. So basically, that is the thing behind being an adaptive immune response. They must first know who they should fight, and that is through the help of the dendritic cells. Okay, so moving on to the what happens to the naive, the activated helper T cell. So it becomes the effector helper T cell and memory helper T cell. So that means that single cell that becomes exposed, they divide, and some differentiate into an effector helper T cell. The effector helper T cells will release cytokines and helps modulate the activity of the activated cytotoxic T cells. And then the other one is the memory helper T cells. Okay, so we have here and then for the activated cytotoxic T cells that that receives the cytokines from the effector helper T cells, it then multiplies and differentiates into the memory cytotoxic T cell and the effector cytotoxic T cells. And the effector cytotoxic T cell is the one that actually fights off these infected cells. So they releases enzymes, perforins that induce the bursting of your infected, actually apoptosis or end bursting or lysis of the infected T cell. Actually, the scenario should be apoptosis, but if that doesn't go, they will lyse the cell and macrophages would eat it. So anyway, that is your cell mediated response. So here is the dendritic cell and meeting up with the naive helper T cell. So that's it for our this part of the lecture. So this is our this also is the end of this part of the lecture for the adaptive immunity.